Welcome, Marina, to this chat. Well, it's a series of chats called PAT Chats with Marina Kirkitadze. We are excited to have you with all your experience during these years. I think it will be a fantastic contribution to the discussions that we're going to have. And the first one, the title is about PAT. And we are going to focus on applications, PAT applications, where PAT works, where it struggles, and why it matters. But first, a bit of introduction about Marina. I guess the colleagues in the PAT environment and community already know Marina, but I'll give a very short introduction. So today, Marina works as a consultant at Biocraft Research Consultancy in Abu Dhabi, and she has 20 years of experience in vaccine industry, acquired at Sanofi and also from her background. So overall, 30 years of experience in biotechnology. Marina got a bachelor's degree in science and physics from the University of Tbilisi. How do you pronounce the university, Marina?
Tbilisi State University, yes, correct.
Perfect. Followed by a PhD in biological sciences and also an MBA from the University of Phoenix in the US. We would like to highlight the experience in process understanding and process optimization that it's going to be topic of our discussions during this series of chats or podcasts with video, and also the experience in the regulatory environment that Marina has. So in getting products through regulatory approvals across multiple jurisdictions, FDA, EMA, and the Health Canada as well. This includes skills on process analytical technology that would be focus of interest here. And we also highlight the 51 peer review papers from Marina and team. Marina is today collaborating with Skill Caper, and this allows her to go deeper in educational technology and going into assess the skills and performance of the personnel and how can we do continuous improvement, and also committed to advance tech bio landscape by connecting global expertise, catalyzing innovations, and promoting sustainable data-driven biotech solutions. So Marina, to start with, perhaps we start slow on what does PAT really means? What do we mean with PAT? And what is PAT and what is not PAT?
Sure. First of all, thank you very much, Anna. It's a pleasure to be here today and have a conversation about PAT, which is a very dear topic, very dear to me because I've worked in that field and I see evolution. So it's really nice to have a kind of discussion today. So I prepared a few slides just to give our viewers some visual. But of course, what is PAT? It's Process Analytical Technology. Why it's also so dear for me? It's because I'm coming from biophysics background since Tbilisi State University. That was my first step in education. And during my career, I always wanted to see biophysical tools converting into the control or monitoring and controlling manufacturing, but also to see it in quality control. And so I think PAT is kind of wonderful bridge that we all kind of gear towards to see that you can produce and also have release at the same time as you manufacture material. So with that, if you allow me, I will just quickly share a presentation like this. Does it work for you?
Yes.
Yeah. So basically, PAT spans the full pharma value chain in R&D, development of the process, like you mentioned, optimization of the process, manufacturing, and also it's aiming for QC. I guess it's still a lot of work to do, but I think we see it best for now in many places, at least in biologicals, it's R&D, and process development, piloting the process. Of course, in spaces like small molecules and biologicals, small molecules especially, it went further and landed into the manufacturing space for blending, drying, coating, and crystallization, and that's where PAT is success story. For biologics, I think where it shines, it's upstream manufacturing. It also supports downstream operation, but it's more challenging at present. But nevertheless, efforts are made, especially with online probes to see it happen. And yes, of course, it doesn't work everywhere. Like you said, what is not PAT? It's when the signal is not strong enough, the signal to noise ratio is not appropriate, then it's not reliable, meaning that, damage happens over CIP or SIP, so cleaning in place or steam in place cycles. So those things affect as well, the coating sometimes happens, the optical window is closed, so we don't get the signal we desire. So that shows that, some technology has to be, further improved or conditions adjusted so that it can perform to its full power. And of course, ultimate topic, as mentioned, it's maturity. So organization is ready for real-time knowledge-driven manufacturing. The readiness of the teams also important. So I don't know if it's sufficient for opening. I have more things here, but should we go on?
Yes, sure. So I would highlight from what you said on the importance of the real time. So we're measuring as we are producing or as the process evolve instead of waiting until the end, right? So would this tell apart PAT from end testing or QC? Would this be a fair differentiation?
Yes, it will be. So with PAT, we want to see real-time measurement, and we want to stay apart or basically shift. I would say the proper word is shift from the end product testing. So with end of product testing, whether it's intermediate even or final product or field product, you still have end result. So at this point, it's very hard to change something, or sometimes regulator do not permit. So when PAT is in place, in real time in process monitoring happens, it transforms the development, and also it allows to set the ranges for the automated adjustment. Basically, all purpose for it is to have this closed loop or feedback loop, so-called, to adjust automatically if the, for example, concentration of substrate or for example, certain component is low or decreasing, it has to be adjusted to a normal level. So these things have been adjusted at time. And then you test if you have, for example, I don't know, infrared probe inserted, and that reports the characteristics of your product. Then, of course, you do it as you go. You can see gradient, right? The intermediates. And then you see closing to the final version of the product at this stage of manufacturing. And then this becomes more proactive. You can first learn, the teams can learn from the process. They can detect variability, deviations faster, and also find the root cause of the event. That accelerates investigations for them as well. So definitely, back to your point, it shifts from the end product testing that we traditionally see, QC or analytical lab, whatever it is at this stage of manufacturing, to real time. When you see it's continuous. So it's more like a movie as opposed to the snapshots in time as photographs. So that's also my paradigm shift. From offline to real-time insight. Overall, it will improve quality because you know more, you have huge data sets. You can see the whole process holistically. And also it's more robust, data-driven process improvements and of course shortens timeline for development as well. So
I love the movie comparison, Marina. I have a question though. So we talk about feedback loop and adjusting as we go along. Are we talking about commercial manufacturing or pre-manufacturing? Because what I'm just wondering is this actually allowed to adjust while a file manufacturing process is ongoing? Can we adjust? Is it allowed?
definitely, it's a perfect question because opens up the situations that I think best apply to research and development, to the development of the product, where you can see during development you adjust and polish the process, so to speak. But if you validate with the range, right, for the process specification, so critical process parameters, then you may see validate within the range for certain parameter. Then yes, this feedback loop would be allowed to adjust. But of course, it takes to work with regulators to demonstrate that this range is robust. It's indeed the process stays within the range, and we all know that sometimes it drifts. So what to do when the drifting happens? How to mitigate that? That will be the question. But for well-known processes, and of course we see it more for small molecules rather than biological, but even for biologicals at the like upstream fermentation stage, or when we use bioreactors, these things can be achieved because you have substrate concentration has to be certain level. For example, you may see metabolites surfacing. It's also, I don't say predictable, but it's measurable at what concentrations these intermediates will stay. So these things can be added into the validation of the process. And the continuous data helps to define what is repeating, one run after another, and what is happening occasionally and should not be used in the validation at all. So with that, I would say R&D is a perfect space for it, for manufacturing. Once you know the process and it's really robust, then yes, we can validate within the range. And then this feedback loop will serve to adjust, to maintain that range. For example, temperature at present and pH in fermentation. That's how it's monitored and adjusted, right? So that's already happening to keep it in certain range, to heat it up so it's not 36.8, it should be 36.9 to, I don't know, 37.2, that range to be carried through entire round of fermentation. So yes, what you know well and where the process is robust, that definitely helps. Yeah.
And the beauty of the power of being able to adjust, so then you can correct and mainly avoid batch failure. That is the ultimate sustainable goal, right?
That's right. Yes, go ahead. I have it someplace mentioned, I think in use cases, but yes, let's go ahead with your next question and I'll try to find the use case that matches it. Yeah.
This drive us also to, okay, what's working versus what's not working currently, what potential hiccups can we have when trying to adopt or implement PAT?
I think what's working is the mature technology. So the sensors have to be mature, the technology has to be mature. And also, on the other hand, the process has to be well understood and well known. So for example, if you use UV visible for concentration tracking and impurity, it's working really well. You can measure concentration related to UVVs. With the same respect, you can do it with infrared, but UVVs is often sufficient to proceed. pH, dissolved oxygen, conductivity, it's present in upstream bioprocessing, but not only. For example, if you happen to produce in-house adjuvant, for example, aluminum-based adjuvants, then pH and conductivity is used in the tanks, of the raw materials for mixing and, synthesizing the aluminum adjuvant in another tank. So with that, you have real-time measurement continuously, and it gives you the full picture of what happens during the mixing. Then, for example, mass flow pressure and temperature, again, for continuous manufacturing, that's often used, and it's reliable because these processes are well known. NIR, we also see it, by the way, in some companies. They use it for the upstream fermentation, upstream bioprocessing, but they also use it for blending to demonstrate uniformity. And of course, for dry formulations, for drying, granulation, and for some liquid processing. So again, the tools are stable, they're well understood, but on the other hand, the process is also well known and well established. So this works well. What else we see? I mentioned already real-time monitoring for upstream fermentation. Raman is there, but not in many places, but it nevertheless finds its way into the upstream. Again, DO, which is dissolved oxygen, pH, capacitance probe gets there as well, is to see the live cells, right, versus non-live, to check that off-gas. Sometimes off-gas analytics link to ELISA results, and there are some efforts towards that to give more meaning how to link. It's still more work needs to be done to see that it's real and robust in terms of modeling, and I think modeling will touch later during the conversation. But again, the process is understood. Small molecule reactions, chemistry well defined, so spectroscopy works well, and also Raman and NIR for reaction endpoints, crystallization, and solvent swaps. So change of solvent, of course, it changes the spectrum. You see, diminishing of one solvent and appearing on the other. Both NIR, Raman, actually can use also infrared, mid-infrared probes to check that. It's very visible, and for small molecules, it's easy to adapt. Easy, I mean, in terms of technical and scientific value. Validation is another topic that we discuss later. So I think you said what's not working.
Yes.
Yes. So here I have inline NMR. Inline NMR, I have to mention, there is, I think this more refers to the 5-field NMR. But I also want to mention there is low-field NMR, and it definitely tries to find its way as a PAT, if not on the manufacturing floor, but maybe in the QC lab, where it can be connected and measured in parallel with the processes, especially processes, for example, like mixing or fermentation. They're usually low processes. So with a low field NMR, you can answer those questions. So we have recent work. We compared near-infrared results with low field NMR results. And Gabriela Gerson, she's the first author on the paper. I think on one of the slides, I have reference to her work. She's currently a graduate student at York University and worked some of it. We started together, so she's closing to her finish line, writing the thesis at present. So that's her recent paper, came out this year. So again, low field NMR definitely has a chance. It has excellent data, but I agree with saying that yes, it is expensive. Yes, it requires expert presence and the knowledge how to use it. Same refers to inline aspect. It's sensitive, but requires expert knowledge and support for this. Another issue is the models, right? We all need to validate the model. and then see whether the process and the points we collect matches that model. And we all know that chemometric heavy models, they often drift. And so what do you do if the model drifts? How to incorporate it into the regulatory landscape, how to show to regulators that if it drifts, what we do to ensure that the product is meeting same specification as claimed. So these things are challenging. These things are not easy to address. And also, biologics itself is both heterogeneity and variability. So on one hand, you may see overlapping spectrum. The signal is low. One compound has the spectrum in the same, or peaks in the same range as another compound. And like many things that, for example, peptides and sugars, they sometimes overlap. Not identically, but they nevertheless mask the spectrum. So it's hard to dissect which one is which. That's where the difficulties actually occur. Even if you have individual spectrum collected for each component, you still have difficulties to translate when they all come together at different concentration, different times. So that limits PAT ability in terms of replacing end of product testing for biologics. But nevertheless, things progress, and yes, and hope is there, and more technologies getting into the PAT space or find its way in manufacturing development, improvement, but also in manufacturing. Another part I want to mention, it's integration and data infrastructure. So the data silos, how the data is collected, how it's narrated, meaning meta files, whether it's all these PAT probes, they are interlinked or they stand alone. That also introduces difficulty because we need the systems that bring them all together as one organism. So another comparison would be, we'll all try to humanize everything, right? Whatever we do. So if we look at the manufacturing as, basically the plant, just like human body is continuously working plant where we interconnected and brain, monitors everything. We want PAT as the actual participants with the software linked together to operate all these complex organisms as one unit. So here, I think SyntiQ, I would mention, Zontal, and Logs, those programs trying to bring it together and close the gaps. And I guess, we can talk about it later, but these platforms definitely give hope to see, when these systems are all intertwined and connected. So.
Perfect. Yeah. And so far, the benefits of implementing PAT seems obvious. You guys explained so many different examples and techniques that we could be using, and then ultimately how beneficial this is. However, we are at 2026. The guide from the FDA was 2004. And yes. We've been many years kind of on lethargy and not maybe completely fully adoption in pharmaceutical industry. What are the adoption barriers that made this quite slow? Maybe.
It's an interesting question. Yeah. You're right. A very interesting question. Why? Because on one hand, there are a lot of proponents for PAT. And of course, if you're on R&D side, or process improvement side, you definitely see the value. But again, manufacturing is more, I don't know what use the word, more strict because they have to comply with the regulator rules, and it's legal environment, in fact, implied. So it's not just technical, scientific, but it's also legal. So what you do, how you prove, right? So the validation becomes a big thing. So what can be validated, I think, goes through and has potential to go through. But overall, validation burden is often the what stops manufacturers or teams to adopt it. Why? Because validation is very costly exercise. And so I think at this point, it's a readiness of the teams to adopt. It's not just technical and scientific, but also readiness of the teams to take ownership. I think so-called long-term ownership when, say, quality assurance, manufacturing, perhaps in interim, the pilot, some people call it manufacturing technology teams, and R&D working together to make it happen. Not just, okay, this is in R&D and it doesn't go through any further because we're not ready, but from the beginning. And it's not just new product, existing product, you're doing improvement, you have new plant, there is an opportunity. Use this opportunity, work together, and implement PAT, even the smallest step. Let's start with pH probes, simple, comparatively, like, to infrared maybe, or Raman, or even low field NMR. Even something very simple, to have it right from the beginning, and then add more. And see if you, in this environment, collaborative environment, identify what are the roadblocks. So if collectively they identified from the beginning, there is a potential that collectively these teams will win the budget to do the validation step, and through that to see PAT come to fruition. So I would say this. So it's really ownership and commitment. And of course they can object. Technically it's not mature enough. All the models are not robust enough, but they drift over time what to do. But all these questions can be, first of all, asked, and then the team can go through whether it's feasible or not, and see if whatever is feasible, maybe out of 10, you have 3 or 4 probes that go through. And this is just enough to control the process. You don't need too many. You maybe just need critical ones. And they can perform well and give you this continuous picture of what happens in this stainless steel tanks, or even in single use. Those are also non-transparent, even though it's polypropylene or something, but it's still not transparent what happens. So this digital world allows us to see it, to give this how to say, a digital vision of what happens inside the process. So this exercise is worth it, even though may appear heavy and too enthusiastic to preach. But the benefit is that overall they can achieve reduction on cost of quality, and also generate the readouts that become release tests. So release as you manufacture. Release as you manufacture. I think that was the call from FDA in 2004, so that the teams can adopt. But I think the technology and the complexity of the biologics somewhat does not allow you to move fast enough. And when there is like weak connections, it often breaks and becomes like energy dissipates. So here we need to carry on with this energy. So it's commitment to get through the entire road to see this happening and materializing on the other end. So that I would say long-term ownership by the teams themselves. That can help to see it through. And I think there are companies, maybe they would be more like CDMOs, but they can be exemplary of introducing PAT and using it in their processes. And Lonza is one of them that uses PAT in their processes, and that gives them significant advantage because they get, let's say, requests to manufacture. So
it is certainly a differentiator and an advantage. And Marina, to conclude now, perhaps what we have discussed, if there was one piece of advice that you would give to those that still relying on snapshots instead of the movie, right? And that are hesitant, perhaps, or thinking about implementing PAT, but they see the mountain to climb. What would you say? What is it that you would advise us?
I would say, the vision of the future is often, well, sometimes it's brave step. And in manufacturing, it's often calculation, it's a budget, it's also regulators, and I think also the essence how they take risk. So in many places, PAT is associated with risk. And usually, you cannot convince people to take risks, but if you help them to find alternative, that if PAT doesn't work, what I gonna do? I need to release my product. How you can help me? If this mitigation embedded in the processes, then there is a better chance for the teams to find solutions and introduce PAT in their system. What I mean is, for example, if PAT doesn't work, what do you have? For example, I have two probes. They sit in fermenter or they sit in the mixing tank, and they both give me readout, and I know that the process happens as it should be. And at the end, I get the product and it matches, the range, the specification range it should have. Fine. What if both probes fail? What remedy do I have? Okay, I will have a small equipment, exact same origins. For example, pH. It's right at the bench, or it's just across in a different room from the manufacturing suite, and I can immediately check what happens. But some things I allowed in manufacturing suites, and they're robust enough to sustain CIP, for example, or cleaning with room cleaning, sterilization. So if you have those offline equipments right next, that's your fallback in case something does not work. I see that if you satisfy the de-risking need of the people involved in manufacturing and knowing like the responsibility they carry to get the product done well and get it, commercialized, then, the other side of the team that, proponent for PAT may have a chance to see, to bring the probes into the manufacturing suite and bring it to fruition. So these things have to be addressed, the risk and the responsibility of the teams versus the novelty, the new process and advancement. Together, I think they can achieve the goal that if PAT works most of the time, they have process and they learn a lot about the process, much more than what they have. But if something doesn't work, they will not have a showstopper. They will have offline testing right at the manufacturing suite, also validated machine. So it's additional investment, additional time, but nevertheless doable. Then they measure the parameter they have to report with this step and they move on. So there is no interruption. Manufacturing process is continuous. And if it happens at the end, so it means that, you have your product release, you have results both by offline and by inline probes. Inline probes do not work. Offline instrument reports the data. So with that, I think to cross the bridge or to climb that mountain, you can climb as people going onto the Everest. It's a camp, right? Small camp on a low field, and then the camp a little bit high, and then the last march when only few people reach the top of the mountain because everybody else supports them. So here the same thing. You have support all the way, and maybe like in this setting, there is a better chance for the probes to see it through into the space. So that's how I would. My suggestion, to respect both sides, because both sides have value, and very often cannot agree because, well, may not hear each other, and but if both needs are satisfied, then you can see success.
Excellent. Fantastic, Marina. And with this, we conclude the first episode of PAT Chats with Marina Kirkitadze. And now we are going to get ready for the second episode that we will dedicate to software and how important software, or not important, is in PAT, but we will find out more in the second episode. So we talk to you then. Thank you very much, Marina. Thank you all.
Thank you, Anna. Thank you all.
Bye-bye. Bye-bye.
Bye.